Alpha-glucosidase is an enzyme with two contrasting identities in modern medicine. Depending on where it operates, it is either a target for suppression or a non-negotiable requirement for survival. Inside the intestines, this enzyme is responsible for cleaving complex carbohydrate chains. It breaks them into individual glucose molecules that enter the bloodstream for energy. In type 2 diabetes, this process happens too efficiently. To prevent post-meal sugar spikes, physicians use phytochemical extracts, like those from the Gymnema sylvestre plant, to block the enzyme's active site and blunt the release of glucose. But while we work to inhibit this enzyme in the gut, we are struggling to replace it inside the cell. This is the reality of Pompe disease. Pompe is a monogenic disorder caused by a total deficiency of acid alpha-glucosidase, or GAA. Without this enzyme operating in the lysosomes, cells lose the ability to clear stored glycogen, leading to progressive muscle wasting. Replacing this enzyme is not as simple as a standard injection. Recombinant GAA can circulate through the blood, but it fails to penetrate the membrane of resistant muscle tissues. To treat Pompe, researchers must structurally engineer the enzyme to force its own absorption. This requirement turns a lysosomal storage disease into an intensive application of targeted pharmacokinetics. In a GAA deficient cell, the problem is mechanical. Glycogen, which should be recycled, simply accumulates, filling the lysosomes until they can no longer function. As the glycogen load increases, the lysosomes swell. They eventually engorge the cytoplasm, physically compressing and deforming the surrounding cellular architecture. This physical crowding halts the cell's internal cleaning process, known as autophagic flux. When the pathways are physically blocked by swollen lysosomes, cellular waste can no longer move. This disruption specifically stops mitophagy, the process of identifying and destroying damaged mitochondria. Without active mitophagy, these damaged mitochondria accumulate and leak oxidative stress into the cell. This oxidative stress damages the mitochondrial membrane potential and depletes the cell's supply of ATP. This chain of events leads directly to the bioenergetic failure observed in skeletal and cardiac muscle. It is not enough to just deliver the enzyme. A therapy must clear enough lysosomal congestion to physically restart the mitochondrial recycling engine. For a systemic therapy to reach the interior of a cell, it must pass through a specific gateway, the CIMPR pathway. The first generation of enzyme replacement therapy, alpha-glucosidase alpha, was designed to use this receptor for cellular uptake. The problem with this first-generation approach is a lack of mannose-6-phosphate, or M6P, tags on the enzyme surface. Without enough of these glowing markers, the enzyme cannot maintain a strong grip on the receptor, eventually drifting away. This weak binding affinity leaves skeletal muscles starving for the enzyme, even when there are high concentrations of the drug in the bloodstream. Second-generation therapies, like alpha-glucosidase alpha, were engineered to fix this specific structural flaw. This enzyme is manufactured with a 15-fold increase in M6P content. By firmly engaging multiple receptors at once, the drug is successfully pulled through the cell membrane. This structural change yields a clinical result. COMET trial data showed a 57-meter improvement in the 6-minute walk test over the first-generation drug. Long-term data also shows stabilization of forced vital capacity, a measure of lung function, over a 145-week extension period. Multiplying the molecular tags on the enzyme directly prevents the typical respiratory and motor decline seen in untreated patients. Even with high receptor affinity, enzymes face a second hurdle, the harsh environment of the human vascular system. Recombinant proteins can denature or lose their shape in the blood, causing them to lose efficacy before they ever reach the target cell. Third-generation strategies address this by using two components, the enzyme alpha-glucosidase alpha and a stabilizer called miglustat. The enzyme optimizes receptor binding, but miglustat serves a different purpose. It acts as a pharmacological chaperone. It physically inserts into the enzyme structure. Acting as a structural brace, miglustat prevents premature breakdown during its journey through the blood. The PROPEL trial demonstrated that this stabilized combination significantly reduces levels of HEX4, a biomarker of glycogen waste, indicating more effective clearance than unstabilized enzymes. Increased receptor affinity only works if the biologic is physically reinforced to survive the journey through the body. As these therapies successfully extend the lives of Pompe patients, long-term survival is revealing complications that were previously obscured. Doctors are now observing cardiac conduction blocks and neurological decline in survivors who have been on standard therapy for years. These symptoms persist because even the best M6P-tagged enzymes cannot penetrate the tight junctions of the blood-brain barrier. They remain trapped in the bloodstream, unable to reach the nervous system. To cross this barrier, researchers are testing a new delivery method that utilizes the transferrin receptor. This involves fusing the GAA enzyme directly to an antibody that targets the transferrin receptor. This fusion protein bypasses the standard cellular pathways. Instead, it triggers a process that actively ferries the drug across the blood-brain barrier and into the central nervous system. Preclinical results in mouse models show that this approach can normalize glycogen levels in the brain and reduce neuroinflammation. To treat the final frontiers of the disease, we have to move beyond the enzyme's native pathways and hijack the body's own vascular transport networks. The evolution of Pompe therapy has moved from basic recombinant proteins to M6P enrichment, chaperone stabilization, and now antibody fusion constructs. Each of these breakthroughs was the result of identifying a specific physiological bottleneck and engineering a way to move through it. This contrasts with the metabolic management of diabetes, which relies on simple inhibition in the gut. Modern molecular medicine is an exercise in spatial mastery. Biologics must be engineered to navigate the exact microgeography of the human body.